Okay, today we're going to be talking about predicting genes in a genomic sequence, with a big focus on open reading frames, Shine-Delgarno sequences, and coding potential. Part 1 of a 4-video series, we are going to be talking about where are the genes in this genome. To learn more about the biology of the phages you isolated in the first semester, we will be analyzing the sequence of their genomes. Recall that you isolated genomic DNA from a high titer lysate of your phage. This was packaged up in a box and shipped to a sequencing facility in order to learn the nucleotide sequence of your phage genome. The result of the sequencing process is a FASTA file, which is a plain text file containing the nucleotide sequence of the phage genome. There is a header line that begins with a greater than symbol, followed by information about the sequence in that FASTA file, such as the name of the phage and the cluster it belongs to. The lines that follow are single letter codes for the nucleotides that make up that DNA sequence. Here's a FASTA file for a phage named Clipper. It has a genome length of 60,558 base pairs. Your goal in this course is to determine from this sequence where the genes begin and end, what the biological function of those genes are, and what this tells us about the biology of the phage. In this lecture, we are going to learn about the processes we use to determine where the genes begin and end in this FASTA sequence. We can start by looking for open reading frames, which include a start and a stop, codon, that are in frame. That means that if you read the nucleotides in sets of 3 nucleotides, or codons, the start codon is in frame with the stop codon. The problem with only looking for open reading frames in a FASTA sequence is there is an enormous number of ORFs in a single phage genome sequence. Let's look at the beginning of the genome of phage Clipper. The FASTA sequence begins at the left arm of the genome, and it is the strand that reads 5' to 3'. The genome, of course, is a double-stranded molecule, and therefore there is also a complementary strand that reads 3' to 5', rather 3' to 5', that is not shown but can be deduced from the 5' to 3' strand. Where are the open reading frames in the 5' to 3' strand? We can read a DNA sequence 6 different ways. In other words, there are 6 different reading frames, and we can find open reading frames in all 6 frames. The first 3 frames are found in the strand that reads 5' to 3' in the left to right direction. If we begin with the first nucleotide, we can read in 3 base pair increments until we find start codons. Start codons may be ATG, GTG, or TTG. We can then look for stop codons that are in frame with these starts. Beginning at nucleotide number 1, the yellow highlighting shows every other 3 base pair sequence in reading frame 1. This frame would have forward transcribed open reading frames. The start codons in this reading frame are shown in green, and the stop codons are shown in red. Note that there is an ATG start and multiple GTG starts. There is a single open reading frame in this region that, of course, has a single stop codon, but there are actually 5 different start sequences that could be the beginning of the protein coding gene. The arrow shows the length of the open reading frame beginning from the first possible ATG start. If we read the same genome sequence beginning with nucleotide number 2, different 3 base pair sequences appear. The starts and the stop of the first reading frame do not exist in this reading frame, And note that new starts and stops appear and new open reading frames, which are indicated by the arrows. And different starts and stops and reading frames appear in the third reading frame. Reading frame number 4 exists in the reverse strand, starting with nucleotide 1, reading in the 5' to 3' direction, but in the right to left direction. For example, the first 3 base pair code reads GGT. Transcription would occur in the reverse direction. Reading frame from nucleotide 2 of the bottom strand is reading frame 5, and here's reading frame 6. This graphic shows the first 1,200 base pairs of the Clipper genome in a program called DNA Master, and it shows all the open reading frames in all 6 frames. Each row represents one of the 6 reading frames, with the top 3 rows showing the forward-oriented reading frames, and the bottom 3 rows representing reverse-oriented reading frames. A reading frame is defined by a stop codon, represented by a line that spans the entire row, and start codons represented by lines that span half of the row. This forward open reading frame, in frame 2, has 14 start codon options, although only one of these starts is the true translational start. There are so many open reading frames, yet they cannot all code for proteins. So how do we predict which of these open reading frames actually code for proteins? The goals of this module are to review the basic components of a gene, to review how translation initiates only at start codons and open reading frames that actually code for proteins, to review the structure of polycistrons or operons, to learn the 4 types of evidence that may indicate the presence of a protein coding gene in a phage genome, and to learn how coding potential is used to detect protein coding genes.